All right, the next section we're going to go over is over incomplete dominance and codominance. Now, we just talked about dominance and recessive alleles compared to each other in the last video. So this will be a short one that kind of runs off of the ideas of the dominant and recessive alleles, but you'll see how they're different. Again, you should be following along and then making sure that you are filling out your own lab, or at least working alongside on a notebook while viewing the lab, since obviously the quality here isn't that good. So for incomplete dominance, the examples we've examined thus far deal with alleles exhibiting complete dominance. One allele completely masks the phenotype of the other allele in that case. Some genes, however, exhibit partial dominance, in which neither allele shows dominance or full dominance. Some plants show partial dominance for flower color. For instance, if a snapdragon homozygous for red flowers is crossed with a snapdragon homozygous for white flowers, the resulting offspring will have pink flowers, a combination of red and white. This is an example of incomplete dominance. So when you think of the term incomplete dominance, the most helpful way to think of it is of blending, or an intermediate spectrum in between the two opposite polar ends. So there's going to be like purebred red flowers and purebred white flowers. And then when you breed a white flower and a red flower together, you're actually going to get this middle pink flower instead. So as you can see here, there's a gradient in shading, and that's what this picture is supposed to be representing. Now, the phenotype of the F1 heterozygotes falls between the phenotypes of the parents. The reason for this is due to the dosage of pigment protein being made and how that affects the phenotype. In many cases, one copy of the dominant allele, which makes 50% of the amount of protein found in a normal homozygous, is enough to provide a normal phenotype. However, in this case, one copy of the dominant allele, again, which produces about 50% of the pigment protein found in a normal homozygous, is not enough to provide the normal phenotype, making the flowers appear less red than the homozygous red parent plant. For problems below, consider R1 equals 1 dose and R2 equals 0 dose. And that just means a pigment dose. When considering only the phenotypes the F1 generation, one might conclude that this is a case of blending, but that is not the case. When you cross F1 pink flowers, they produce red, pink, and white flowered plants in the traditional 1-to-1 phenotypic ratio, disproving the idea of blending. Now, earlier when I said blending, that is in order to recognize that there is a potential for the case of incomplete dominance. Obviously, they don't want you using the actual term blending because the term incomplete dominance is more correct, because when you go down the generations, you still see the 1-to-2-to-1 phenotypic ratio. So we'll do a practice problem now, and we're going to do the incomplete dominance in snapdragons. In snapdragons, the allele for red flowers is R1, R1, and is incompletely dominant to white flowers, R2, R2. The heterozygous condition results in pink flowers. A gardener crosses a red snapdragon with a white one. What are the chances that the offspring will resemble neither parent? Right? The questions are a little bit tricky sometimes because they'll throw in words like either or neither. And in this case, it means that what are the chances that the offspring will not be white and not be red? And in that case, they clarified it by saying, in that case, it'd be pink, right? So we're going to be writing out the cross described in the problem. And they said a red snapdragon with a white one. So we know red is R1, R1. And the cross is with an R2, R2, because we know that that's the white one. So we'll fill in the Punnett square right here. It doesn't really matter which one is female or male. So we'll just put the R1, R1 here, and then the R2 and the R2 here. And we'll fill it in. So here we get an R1, R2. Here we get an R1, R2 likewise. Here we get an R1, R2. And lastly, we get another R1, R2. So under each genotype, write the offspring's phenotype as well. So we know that R1 is 50% red and R2 is the white. So when the white and the red are together, they're going to be pink. This is R1, R2 again, so it's pink. This one over here is R1, R2, so it's pink. And the last one is also pink. So we'll cross two of the F1 progeny, which means we're going to cross two found in here in the boxes, right? That means we're going to be crossing an R1, R2, and an R1, R2. Okay, so we're going to write them out. And we're going to complete the Punnett square. Okay. Under each genotype, write that offspring's phenotype as well. So we know R1 is red. We know that R1 is pink. We know that R1 again is pink. And lastly, we note that R2 is white. This cross right here is what they were talking about, where when you do the parental cross with a parental generation, so a homozygous dominant and a homozygous dominant, or in this case, a homozygous incomplete dominant, for both, then you get this weird blended intermediate. But when you cross those intermediates again, you still get that 1:2 to 1 ratio of the either ends of the spectrum and then the intermediate. So that's why they were saying this isn't technically blending, because the offspring don't just make more pink flowers. They, even though the parents are pink in the F1, the F2 generation actually ends up with a variation of flowers instead of just being more pink. Alrighty, so the next practice problem we're going to do is incomplete dominance in human hair. And this is the most studied example of incomplete dominance in humans. Inheriting an allele for curly hair from one parent and an allele for straight hair from one parent will give a hair texture that is the intermediate of the two. Sorry, that was the reading right here at the top. So here's the picture that kind of represents that, right? There's straight hair, which is C2, C2, wavy hair, which is C1, C2, and then really curly hair, which is C1, technically that would be C1, C1. So make sure that that is corrected to C1, C1. So 2 wavy-haired people marry and have 4 children. Assuming that the family shows the expected statistically predicted pattern, how many would you expect to be curly-haired, how many wavy-haired, and how many straight-haired? Suppose you examine the actual children and discover that 2 of the 4 have curly hair. How did this happen, and explain? Okay, so you're going to do the cross here. I will set it up for you, and then you'll do it on your own. You'll answer questions 1, 2, and 3 on the pause, and then I will explain 4, and then you'll answer after I explain 4. So 2 wavy-haired people marry and have 4 children. Wavy hair is C1, C2. So we're going to write the cross out, C1, C2, crossed with C1, C2. And so the mom is going to have a C1 and a C2, and the dad is going to also have a C1 and a C2. So you fill out this Punnett square and answer 1, 2, and 3. Remember, the genotype leads into the phenotype here. So you have to do the genotypes first to be able to decide the phenotypes. So I want you to pause and go ahead and do that. Okay, now look at number 4 again. It says, suppose you examine the actual children and discover that 2 of the 4 have curly hair. How did this happen, and explain? Well, we know that each one of these boxes, right? If you have C1, 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 C2, C1, C2, and then C2, C2. Well, if you look here, C1, C1 means that 1 out of 4 chance of, each time there is a kid born, there is a 1 out of 4 or 25% chance that that kid will have curly hair. On the other hand, each time any kid is born, there's a 50% chance that that one kid will have wavy hair. And each time a kid is born, there is a 25% chance that that one kid will have straight hair. Okay. If you notice the repetition in what I've said, it's not saying that when there are 4 kids, you're definitely going to have a 1:2 to 1 ratio of hair type. It just means that whenever one kid is born, the dice is rolled for the chance where 25% of the time, or they have a 25% chance of having either straight hair or curly hair. And then there's also a 50% chance that they could just have the intermediate wavy hair. Okay, so it's not about out of how many kids you have, that ratio needs to match this. It's about each time one kid is born, there is a certain percentage of chances that they can be this phenotype, or this phenotype. Okay, so codominance is the next thing we're going to go over in this video. And codominance is similar to incomplete dominance in that there is an intermediate phenotype. However, with codominance, the 2 traits showing, they both appear in the offspring, and they appear in different parts on the plant or the animal. With codominance in flowers, the resulting offspring between pink and white flowers would not actually be pink. Instead, they would be red with white spots or white with red spots. The result of both colors being codominant. Okay, so that means that they're both dominant, so they're both going to show up and they're not going to mix together. The codominant alleles encode proteins that function slightly different from each other, In a heterozygote, the function of the protein affects the phenotype uniquely. So when you think of codominance, the easiest way to remember them is when you see patches or spots. Co means that they both show. So they're not blending. They both show in their full capacity, but on different patches. Okay, so rhododendrons are another type of flower, and instead of showing incomplete dominance, they show codominance, which means that they end up actually having patches where you can see there's darker colors and lighter colors on the petals. In rhododendrons, red flowers have the genotype capital R1, and white flowers have the genotype capital R2. The heterozygous genotype capital R1 produces flowers that are both red and white. You cross a red flowered rhododendron with a red and white rhododendron. Okay, so we're writing out the cross. You're crossing a red one, which means that it's going to be R1. And then the cross is with a red and white, which means that it has to show both, which means it is going to be a heterozygous one, which is R1, R2. And let's write that out here. Okay, you're going to fill in the Punnett square. Okay, you're going to determine the phenotypic ratio. So right now we have a 1:1 ratio. And this is red, and this is red and white colored. So right here, we have a 1:1 ratio of red to red and white, which would be your phenotypic ratio. And what is the probability that this cross results in plants producing red flowers? Well, you figure that out based on the percentages that they're shown in here. Write that down on number 4. All right, so a practice problem would be codominance in humans, which is for sickle cell disease. So hemoglobin is an oxygen-binding protein in red blood cells. Sickle cell disease is caused by a misfolded hemoglobin protein that tends to clump up and cause malformed red blood cells. The clumps of misfolded hemoglobin can clog blood vessels, causing organ damage or even death. The hemoglobin gene is represented by HB. The normal allele is HBA, and the sickle cell allele is HBS. Sickle cell is an example of codominance in humans. So this is a picture of a normal shaped red blood cell. There's a little divot right here in the middle where it's kind of like a donut that you flattened in the middle, jelly donut, or it's kind of just like a bun where you squish it down in the middle. So if the cross section is just kind of squished down in the middle. A sickle cell is a very different shape because it literally looks like a sickle or like a moon. Okay. An adult with homozygous normal hemoglobin would have the genotype HBA. We know it's both A because it's homozygous and normal. An adult with sickle cell disease would have the genotype, they have to have HBS. Right? To have the full disease, you have to have both alleles as S. An adult who is heterozygous and has sickle cell trait would have the genotype HBA, because they are heterozygous. A heterozygote is sometimes referred to as a carrier because the person has no signs of the disorder, but he or she can pass the sickled hemoglobin allele to his or her offspring. So carrier is important vocab because it means that they carry the recessive or the disease in many cases without showing. And that's important because sometimes you'll think that in a family tree, a disease appears out of nowhere, but it just turns out that there's been many generations of carriers for a long time. Okay. And then all of a sudden it met with the recessive of the other person who might have also been a carrier. And then a kid is born with a disease and people are like, oh my God, what happened? So 2 adults with sickle cell trait mate. Using a Punnett square, determine the genotypes and the phenotypes of the possible offspring. So we're going to actually do this. They have a sickle cell trait, right? A trait is a characteristic. And we're going to actually say that this means that this is the condition that they show. So if they show this condition, we're going to be talking about genotypes here crossing, right? And that would be an HBS crossing with an HBS. Sorry. This would be HBS, and HBS, because obviously they carry each. Sorry, I got confused with the letters. But the HB is actually representing the whole allele. So it's actually 2 letters instead of just one base letter. So here we'd have an HBS and an HBS. And then over here we have an HBS as well, and HBS. HB probably just stands for hemoglobin. Okay, then you finish your cross and then discover and tell me what are the genotypes and what are the phenotypes of the possible offspring, and write that on the side so that I can get that. I will be checking your answers in your turned in notes.